Control, back deck, that's Atlanta heading into the water.
Control copies.
That's Hercules. Dive.
This is an audio slate for dive Hotel 2100. UTC time is 020712. Mark.
Control deck, can we get the dive screen down here on the social deck?
Copy. Does that look right, deck?
A black screen down here.
Deck control, video's working on it.
Copies. Control deck, in the meantime, can you guys keep an eye on the winch, and if anything starts looking funky, just yell, I'll stop.
Roger.
Control, back deck, that's all stop, 50 meters, control is being sent to the van.
Control copies. Deck, are you okay with holding position? Ship position.
Deck is fine with holding position. We are relieving off to you guys.
Control copies.
Front row, are you ready for us to be on? Or do you need some more time?
I think we're fine.
You think you're fine?
Yes.
I'm good.
Okay. So, half a day, I'm Shannon Salim. I'm the Science Communication Fellow on the EV Nautilus. Today we are descending down to about 550 meters, and we will be doing a shallow seamount on the West Marianas Ridge. This is one that has been previously unexplored. We will be looking for flora and fauna and taking some samples, as well as we will be doing more transects today, which is where we go down at a steady speed and depth without any zooming, and we take some photo data so that other people can do studies on the area. We'll let you know when those are coming up. So for now, can we start in the front row today and do some introductions for the 12 to 4?
Yeah. Hi, everyone. My name is Isabella Monaco. I am one of the ROV navigators on Expedition NA-171. I am a contractor with Ocean Exploration Trust.
Michael Hannaford, I'm the Herc pilot on the 12 to 4 watch, and I'm a contractor and just here for 171.
I'm Annabelle Baker. I'm the Atalanta pilot on the 12 to 4 NA-171 Matinghan expedition. And I'm a contractor and also a student at OSU. Thanks, Dan.
My name is Bryn Hodge. I'm the video engineer intern for this expedition. I'm a student at Florida State College of Motion Picture Arts.
Hey, can we turn on WCA if the temperature is okay, please?
Sure, we good to turn on WCA? Roger. 1 second. WCA coming on. Upper, mid, and lower. Those should take a minute to boot up. Thank you. Of course.
You're not talking.
Hello. I know how to run the controls for my microphone. Jeff Beeson, Oregon State University, assistant professor of marine geology, watch leader. And what time is it? Oh, 12 to 4.
Hello.
$300.
Someone's asking again how deep we're going. We're going down to 550 meters. And just to let you know, this is a different seamount than yesterday. We are not just going up a different direction.
Yep, we transited last night and then surveyed another seamount in the West Mariana Ridge and are exploring this one. So hopefully we get a little bit of a comparison between two seamounts that get very shallow. We saw some sub-100 meter, sub-50 meter pings in the mapping last night. And we're going up the south side.
Hopefully we don't have current like yesterday and aren't as affected by that.
Yeah, the currents were challenging.
We're already down to 329 meters.
I'll introduce myself in a second when I'm done setting up. Sorry.
Take your time.
The pictures of the seamount on the dive plan were like really incredible. Looked super, super shallow.
Yeah, I'm kind of excited about this one. I kind of hope we get up into those higher waypoints. We are starting and can't quite tell how many we have, 5, 6. I think there's like 10 waypoints.
I see a waypoint 9. I think it may, I don't know if I see a 10. These first, I think most of the first, most of the dive until they get to, until Herc gets to the top of the seamount is going to be pretty steep. I think they measured, around 30-degree slopes for most of the beginning of the dive between waypoints 1, 2, 3, and 4.
I do not believe that this is the seamount that Bill was talking about yesterday. This is one that is unexplored, so I don't think that we are going to see the black smoker vents that Bill was talking about yesterday. These are a little bit too shallow.
So tune in tomorrow.
Tomorrow is the black smokers?
Vents.
Yeah.
That is in the Hoffaday.
Oh, Hoffaday.
Vent field.
And just to clarify, in our stream yesterday, everyone was calling it half a day. It is actually Hoffaday, H-A-F-A-A-D-A-I, and that is how you say hello in Chamorro from Guam and the Northern Marianas.
Is it 2 words?
It is 2 words.
Hoffaday?
Hoffaday.
Okay, Hoffaday. I like it. So, is it the equivalent of like the Aloha?
Yes, it is.
Okay.
It is.
That's awesome.
Very nice.
Also, camera is set up now, so now I can say hello. I am Sierra Landreth. I am a scientist. I'm talking like a robot. I'm from Florida State University, graduate school student studying deep sea megafauna like coral and sponges on the 12 to 4.
I'm Evie. I'm a master's student at the University of Rhode Island, Graduate School of Oceanography. I study rocks, more specifically volcanic rocks.
Nice.
And I am the data logger on the 12 to 4.
And I think she's a little upset. We changed our grabber hands to coral cutters from the bigger ones for rocks.
It's all right.
We can have that on one dive, I think. Jacob will be okay with that.
We'll be fine.
Shannon, did you see the rock we got yesterday?
No, I did not.
Oh, it's like, it was amazing. Hold on.
It was cool looking.
What made it amazing?
Because it like was all black on the outside, so we were like, oh, maybe it's just a volcanic rock. And then we cut it open on the saw, and it was all carbonate, which is white on the inside.
Yeah.
Oh, that is really cool.
Yeah.
That goes to show you, you need a clean face to know what type of rock it is. You can't just base off of the video, because it's got the
marin,
the crust, it's got all
you can see that far away.
I know.
That's what it looks
can
like.
see the difference.
Look at the rock.
Yeah.
Okay, my hair's crazy today.
So this should be a fairly short amount of time down, so you don't have to hear us banter over blue water the whole time.
No.
We don't have to see another one of Sierra's magic tricks.
What did you say?
I said yay.
Oh.
Someone said something.
I said we don't have to see any more of Sierra's magic tricks.
Yeah.
Well, Sierra already has a shout out. Whoop, whoop, 12 to 4 here for hashtag Team Sierra.
Team Sierra.
Team Sierra,
or
yeah.
my Coral Collective. So
the Coral
affectionately
Collective.
named.
Coral Collective, have you been working on that podcast?
No, but I have been working on the coral samples, which is why I'm here, not the podcast.
Well, okay.
Got some
Coral
cool
samples
ones
first, podcast second.
Yeah.
The Coral Collective sounds cool, though.
My Coral Collective. I feel like I should make some stickers. Like make a cute little design and sell some stickers, or just send them to people that are interested. And then the rock people could make rock stickers. And then you could be Team Rock.
Oh, Team Rock?
Team Rock. Rocks rock.
I'm going to go ahead and restart the Hotel West.
So the question that we always start with, and I think that we're kind of expecting more coral than rock.
3 beams.
Sarah, what do you think you'll see or
You've got
want
altimeter?
to see in this? Want to see is?
Want to see. Well,
Yes.
yesterday I was really excited about our cold water coral reefs that we saw. They did look pretty unhappy, I will say. Lots of them are already either rubble at the bottom of the seamount or very, very covered in sediment and substrate and kind of on its way to become carbonate in the future. So it'd be cool to see more reefs and be able to get some samples of those. We got, I think, 2 different reef forming coral yesterday, one of which I had never seen before, which was kind of cool. And the other one came with some brittle stars, and we think that one might be Desmophyllum pertusa. So yeah, I got to take a look at those, and then we got some cool octocorals yesterday.
And we got the sea biscuits yesterday.
Yeah, we got sea biscuits.
Sea biscuit.
Someone on here says they're working on a sticker of the sea biscuit from yesterday winning a derby.
Winning a derby, that's cute.
Yeah, very nice.
We've got 58.
Good reference to sea biscuit.
Yes.
I'm going to start slowing down. Oh, keep going? Okay.
I feel like stickers in general are just a great way to show deep sea creatures and get people asking questions, because how many times do you have like a water bottle and someone's like, oh, where's that sticker from? And then you're like, glad you asked.
I'm excited to get our expedition stickers when we get back to Guam.
Oh, yeah.
Our next SCF shell has them. They are already in Guam.
Exciting.
Yes. And I do actually have a shout out for Miss Celine on this one. So it's not a hashtag Team Celine, but we got a shout out.
Roger, I'll stop.
We're getting close.
Terrain on the 30.
Annabelle, look at all the stuff on the Atalanta camera. Look at all the little fish.
Little squigglies.
Yeah.
So cute.
Here, I'll put my tilt down a little bit. Maybe we'll see some more.
Seeing some returns on the sonars. That might be a wall.
Yeah, it does look like quite the wall.
Yep, this is going to be, we're going up slope.
That's your wall.
You're my wonder wall.
Oh no.
I can't help it.
Get out of here.
I just love Oasis so much.
Me too, Jeff. I'm with you. This is now you should start singing.
Lots of marine snow.
Yeah, that's a good sign.
Yeah.
Isabel, if you want to move us more to the east to line up, you can go ahead and move the ship again now if you like.
Oh, that's a shrimp, I think.
Yeah, it did look like it.
We got things swimming around.
Little fishy there.
Would you like me to follow you down?
Oh, sea floors coming into view.
No, stay where you are.
Roger.
It's a big wall.
Is it the sea floor or the wall?
I think that's the wall.
It's not much of a floor. It's more of a vertical surface.
It's the wall. So you talk about Oasis, I think Pink Floyd when I think the wall.
You know what? All we are?
That's
just
the
another
album,
brick in the wall?
The wall, yeah.
Yeah.
So they say that we need some merch on these expeditions. They vote for deep sea creature plushies. I'd vote for a headless chicken monster for that.
Oh yeah, that'd be a good one.
And for Jacob, maybe a rock or two. You can
He's so
tell that
he's
people
so easy
listen
to
to
shop.
us
Yeah.
multiple days in a row.
Perchance a rock?
Jacob is so easy to shop for.
What do you want for your birthday?
How heavy do you think his luggage is going to be on the way back?
That's a good question. I don't know. How many of these rocks is he taking home personally?
Okay, just turn into the slope there, Annabelle, so you get it normal to your view.
It's a lot of rocks.
Evie's going to take some too, just to help him out. They're at the same college.
I don't got room in my bags. Are
Yeah,
you kidding
bag
me?
allowance.
You're taking United. They give
All
you
the rocks. get in these boxes get shipped to our school.
Yeah, they don't.
So we don't even
I think
gotta
that's already
pay
paid
for
It just depends on how long you want to wait for them to get
And
there.
shipment
That's
from Guam
true.
is expensive, but it's going by a boat, girl. It's going to take a while.
Yeah.
I'm okay with that. These aren't for my project.
Okay.
I did possibly take a rock.
You
but-
are going to have to tell us about your rocks at some point.
Oh no.
You've opened that.
Oh,
You've opened that door
Oh no.
when you said you like rocks.
Did you
but
not
not
know
these
that I like
rocks.
rocks?
I assumed, but I don't want to.
You didn't want to.
Don't want to just blurt it out.
No.
And Jeff just brings home stinky water.
Stinky water that then I give to the chemists. Here, enjoy your stinky water. Seeing some sand,
okay, come on down.
some ripples, some
right
current
there.
ripples.
Yep.
I'm already thinking there's more sediment here.
What
Oh.
are you?
That big long.
Neat.
Looks like a south.
Ooh.
I see
Corals already?
coral.
That's a,
Oh,
that's a big
yes, that's
That's
a
a big
great
rock.
sign.
Oh my gosh, guys, coral.
Is that a coral or like a sea fan?
Sea fans are a type of coral.
Oh, okay.
But coral.
So both.
Yes. Yes and yes. And what's the, I see little white, or you got maybe some squirrel lobsters maybe?
Spot lobster.
I don't know, maybe. I can't see. They look like it though.
You have to get closer. We're kind of still moseying down.
Shrimps maybe.
Our step is finishing out.
Roger.
No, we're almost down. 504, 551 meters.
Okay, come down a bit more.
I'm gonna come off auto iris. It's gonna be a little.
Okay, go ahead and zoom in when you're ready.
Got it.
Just fix that for you.
Thank you. Okay, it's gonna go black for a little bit.
Roger.
In Atlanta.
I'm all done.
Thank you.
So someone asked about the lasers. You can't see them up there right now, but when you do, they measure out 10 centimeters so that we're able to tell the different sizes of what we're seeing down below.
It can be tricky to know the size of things with the cameras. So the lasers are a nice check of, they're 10 centimeters. So when they hit something.
All right, we're ready.
We're ready.
All
okay.
right.
Well, let's start looking around and heading to waypoint 2.
Okay.
Waypoint 1 is slightly to the east, but it's not very far. You want to just proceed to 2?
Just proceed to 2.
Yeah.
Copy.
And if you're seeing on the camera. If you feel free to have a little bit of liberty to just, if there's some neat stuff, zooming in on it, or if there's corals or animals, don't.
Sure thing.
Don't wait.
Woohoo.
If it's something neat.
That's what I want to hear.
We get to just explore for a while.
Yeah.
What waypoint do we have to do the transect? Do we have a while?
2 to 3.
2 to 3.
Can we zoom in on the high pack
Yes.
a little closer?
Thank you.
2 to 3 and 4 to 5.
Okay.
Awesome.
Yeah, that sonar is definitely lighting up.
High pack circuit.
Is now a good time for gauges?
Sure, yep.
Yeah, I like this one. High pack climate. No, high pack circuit. Okay. I'll say so far, looks about the same.
Carbonate. Our good friend carbonate.
Our good friend, carbonate and sediment.
What more could you want? Did you say lava?
Lava would be okay. Not, you know.
There's a lot of sediment here. Just a big old dump of sand.
Yeah, that's been problematic. So you have it on the gauge, HPU 3000, and then you have the res there, right?
Have our interloper.
If you can try and refresh it. I don't know what else to tell you to do with it.
It's very pale if it is alive.
Oh.
Oh, there's a little bit of living right there. Hey.
Sure, go ahead, yeah.
Very pretty. Oh, we got our urchin friends already too.
Oh,
he's being blown away. Bye.
So Jeff,
zoom in there.
Is this active or more ancient seabed like yesterday?
Ancient seabed like yesterday. This is a very similar seamount on the remnant arc or Western Mariana Ridge. This is the old arc from 8 to 10 million years ago when this was the active volcanic arc. And then with back arc spreading, initiating, pushed us, started pushing the remnant arc to the west, moved it outside of the subduction zone kind of factory where you get volcanism and lava produced there. Then it went cold, and now we have these remnant seamounts that used to be active volcanoes and now have had millions of years to get carbonate formation and erode. So it's a really nice, this is one of our 3, maybe 4 if you count our abyssal plain or Pacific seamount environments that we've visited while on this expedition. But that means there's been a long time for reef building and potentially habitat and ecosystem that isn't interrupted by the occasional eruption or lava flow.
So Mike, our audience wants to know if they find a rock for Jacob, could the left arm of Herc pick it up?
Yeah, it can, but it's a bit clumsy. We would, we can't, if it was loose, we could still get it with the one with the coral cutters on it.
Awesome.
We could pick it up with the left arm, but it'd be hard to get it in a box.
Yeah, I see a Sako. Yeah, that looked like a coralliid to me. Sako saying maybe a hemicorallium. It was very pink.
So we have an interloper with us again today. Would you like to introduce yourself?
Oh, why, hello. Yes, it is I, the interloper. My name is Kelsey McClellan, and I am faculty over at the Northern Marianas College. And I am honored to be sitting with the 12 to 4 today. Thank you guys for having me.
It's an honor to have you, Kelsey.
It's an honor to have you.
It's nice to have you back.
Guys. Thanks.
She's just here to monitor our transects.
Never.
Make sure we're doing what we're supposed to.
Well, here's hoping for lots of things to identify on those transects.
Woo! That would be cool.
That would be cool. And to confirm for you, transects are occurring between 2 and 3. Waypoint 2 and 3 is the first one.
What's the depth of 2 and 3?
I'll get you the exact information.
Your gem.
The depth for 2 is 510. For 3, it's 411.
Perfect.
So about 100 meters of altitude, depth change.
Yes, and I think that the distance between the 2 points is 150.
That's excellent. Why I was also looking at that is just from the depth that we took transects at the last site as well.
Okay.
And the depth of one of the transects was around 500 meters and the other was around 280 starting, so that's good.
Nice.
Lovely.
Yeah. It's as though they've planned it.
Almost.
Crazy how that works. Yeah, probably brown. That's very cool. Very funky. Oh, there's something perched on top there too. A couple of coral friends, sponges. Now, if I see a lot of pink coral, I think I'll also be pretty excited.
There's a pink one up there.
Yeah. And a little swat lobster.
Oh, what's on top of the rock?
See him sitting down there too. Big old lobster.
That's a cool little community on the smaller rock.
Can we get a zoom up here?
Yeah, it's getting a bit closer here.
Moving into position.
Thank you.
There's a thing sticking off the edge of that one on the right.
Yeah, on the right. Yeah, these are a bunch of different kinds of coral.
They've got themselves on that nice rock.
Yep.
Good
Okay,
location.
video, go ahead, zoom in.
Thank you.
Oh, yeah, look at, so that's actually looks like a basket star right there.
That's really cool, though.
Hanging out on that coral.
I like all the
Zoom in on
different
these right here.
colors.
Yeah, he's gorgeous.
So you think the pink one's basket coral?
No, it's a basket star.
Oh, it looks like
Basket
there's
star,
one.
okay.
Yeah, it's a type of echinoderm, so kind of like a sea star. And then this might be a Paramuricidae or some kind of a Canthogorgia there on the right, and you can see some claws sticking out.
What is off to the left? Is that just a little white rock,
is that a
or
shrimp
is that
right?
Yeah, there's some shrimp
Oh.
and a little hermit crab.
Oh, a hermit crab.
Oh,
nice.
wow.
Look at him.
A cool little community on the rock. Oh,
Oh, there's like a ton
look
of
at
them.
that. Yeah. Seems like this is a good spot for food.
Yeah, a couple hermit crabs, a couple shrimp.
Like a little house, little family.
Yep.
So more than likely these guys are,
Okay, full wide.
A, they have a good place to get food, or B, these coral are kind of a good little hidey spot for them, so a good little habitat for them to settle on.
I think it's so cool when the stars like wrap around the coral like that.
Yeah, so you get a lot of those basket stars, and then a lot of time you get brittle stars or the ophiuriorids. And they like to hang out in the branches of the coral.
Yeah, they blend in too.
Mm-hmm.
Can't notice them until you get really close.
Coming up, just got an eye on that terrain there.
So I guess we
yeah,
have a holiday we would all like. I guess
Going
on Sunday it's
Auto-Iris
World
right
Snail
now.
Day.
Oh. Happy Snail Day.
Yeah.
Oh, what's all of that?
Some more corals.
Just Control-Alt-X and then
It's quite a
should
few.
be on a later page, but it should say HiPAC 1.
Little coral community on the spot.
You're welcome. It's okay.
Look at that one on top. Ah, so pretty.
Sierra's in her element today.
I know, I'm having a good time.
Some squatties down there too.
Yeah.
Roger.
Oh, this one's huge. This looks like it could be some kind of... I'll just see close up. Very pink. Very bright pink.
Do you need a zoom on it, or are you okay?
Yeah, I think they're going to get a zoom.
Go ahead and zoom in there.
Gorgeous.
Thank you.
Yeah, this is definitely a bamboo coral right here, maybe an Acanella, but you can see these little branches right here, these little black nodes. That's kind of how we distinct or characterize these guys, and all throughout, if you were to look really, really close up, you would probably find a little fauna sitting in there too.
And we're going close up.
Woo!
So what makes it an octocoral?
Good question. So, see these, you can't really see the polyps open. Oh, look at them right there.
Oh.
Told you these hiding. If they were to open up, you would be able to count 8 little tentacles around each polyp. So that's why we put them in the Octocoralia group. If it's 6 or like more, like multiples of 6, then we would stick it in the Hexacoralia group. So it's just the number of tentacles around the polyp. And these guys are a type of bamboo coral, so they're in the family Keratoexydidae. And those are the ones that sometimes you'll also see, they just look like one singular branch. They're in the same family. So there's just a bunch of different kinds of morphology of bamboo coral.
Okay, full wide.
Is that a squatty or a regular crab?
Looks like a squatty to me.
Squatty?
Mm-hmm. Thank you. Gorgeous shot.
So we're about 545 meters.
Yeah, so if we collect coral today, probably looking for something we haven't collected yet. So I think we got an Acanella yesterday.
Okay.
So I probably don't need that guy again. Asako agrees with me that it's a bamboo. Thank you, Asako.
I haven't got a hit. Oh, there you go.
Some fish up ahead.
Can you refix her
Yep.
there?
Looks a lot like the sediment we had yesterday.
Yes, very similar.
some larger rocks here though, some bigger boulders in between all the sediment. We got some more coral, little friends.
That's a great view of Herc from the Atalanta cam too.
Yeah, it's good.
Yeah, it is. I'll highlight that.
All right. I think we're still moving to waypoint 2.
So, Jeff, do you want to talk about if we've ever seen bioluminescence down here and how that would work?
Oh. I used the word bio in it. And I'm a marine geologist.
Oh, yeah, that's right. I should have lobbed that to Sierra. I'm sorry. So, Sierra.
You lobbed it to me and I quickly passed.
Okay. My fault.
But I would love to hear about bioluminescence.
So in the deep sea, in general, yeah, there's a fair bit of different kinds of fish.
Oh wait, hey, can I stop you real quick? We are about to get on waypoint 2 to start the transect. Do we need to, are we good to our run in and our starting?
Yeah, we're just getting set up here now, so hopefully as we go through it, we'll be on track.
Okay, if you give the shout out when when we pass through waypoint 2, we can log it as the start of the transit.
Definitely.
Okay, you can now. Sorry,
Or
I just was like, oh, we're almost here.
20 meters out from crossing into waypoint 2.
So bioluminescence.
Birds are hard.
There are fish in the ocean that bioluminesce. A lot of people like to talk about anglerfish and things like dragonfish that bioluminesce, so either their lures or the sides of them. But unfortunately, it's very hard to pick up, which is regular, even really good quality cameras can sometimes not pick them up in the ocean because it's so faint. So a lot of times you need some kind of filter or some kind of lens. And a lot of people don't know, but actually coral can also bioluminesce. There's some black corals that have been shown
Our
to
bearing's
as
going to
well.
be 010.
So not fully understood why they do this. Could be maybe to attract plankton, zooplankton closer. But it's a phenomenon that happens. It's just really hard to catch with our cameras.
Can you talk about bioluminescence with the coral when they're spawning? Where they'll
point 2, right?
I've seen it in
yes.
I was in Roatan, Honduras during the time when the corals were releasing I guess spawning larvae, however they send the more coral into the water, and it was did a night dive and it was like swimming through stars. It was quite trippy, but really cool. And apparently it happens in just one of the months of the year.
Awesome.
I don't know if it was during coral spawning, but at one point my husband and I were diving in Guam, and we had our DPVs with us, those little underwater scooters.
Herc is on waypoint 2.
Dive propulsion vehicles. And as we went through the water, it was just following us along, and it was really awesome bioluminescence.
Very cool. I have been on the back, I've been on a vessel off of South Africa.
We're starting at 4.
Seen the bioluminescence track in the wake of the boat behind it, which is really neat.
So just to let everyone know, we are going to be doing a transect. This will mean that we will be going at a steady pace and at a steady height. And unless we see something really, really, really over the top awesome, we will not be stopping to zoom in.
Yeah.
This is for a scientific study.
Yeah, you can think of this a little bit like a sample. We're doing a video sample of 150 meters with the constant zoom, constant speed, and roughly the same track.
Are we doing 150 or 200
I think
meters?
it's
150.
Okay.
That's what was the waypoint distances.
Yes.
Roger.
If you want 200, we could just continue past waypoint 3 for another 50 meters.
I think that there was some concern about the distance in terms of like the entire
50 meters was concern?
Additional 50 meters after transect 2.
Oh, okay.
Yeah.
After transect 2.
Like for after waypoint 5, you mean?
Yeah, after
It'd be
waypoint
kind of too
5.
steep or something?
Yeah.
Okay.
So then that limits transect 1 to being just the same at a 150?
I mean, realistically, we would have everything be,
Video, do you mind
you know,
opening
the same
iris on
distance,
Atalanta
so...
a little bit?
Yeah.
So 150 and 150 at this one?
Thank you.
Okay.
Correct. And then I'll trim 150, the other transects down to 150
the
as well.
ones from yesterday.
Yeah.
Sounds good.
There we go. Trying to get it where it's not flickering.
Someone asked what would be an example of over-the-top awesome. I think
man.
yesterday we determined it's a giraffe.
I really want to still see an octopus.
Yes,
If
an
I
octopus
think
would.
an octopus would, we would look at the octopus.
That would be awesome.
Or at least come back and look at it.
That would be stop-worthy.
Although we did see urchins on yesterday's transect.
Yeah, that was, this was transect 3?
Yeah, it's one of the
Yeah,
last ones.
I think it was the
Yeah,
third
we
one.
definitely hit that transition to like, oh, okay, there's a lot more stuff here now.
Yeah, that's kind of what we're working with right now, is working our way up the seamount.
Upslope, we've already, what did we start at? We 530 or something? We've gone up about 30, 40 meters?
We started at 550.
Oh,
so
yeah, we're already 50 meters up, 60 almost.
yeah, 60 meters up. So what happens if you happen to come across a creature that is in your way in a transect? Would you deviate or stop? I think it would be kind of hard to find something tall enough
Well,
to...
we're off the seafloor about probably 2 to 3 meters, and we're obviously not going to run into a wall or a rock,
Something right down there.
but I suspect we won't have any issue with. It would be a really interesting creature for it to be in our way.
Yeah, that's
that
what
could
I was
block
thinking.
us, which would then rise to the we should stop and look at it.
Oh, there's a little starfish down there.
Oh, that's good.
Saw him
Starfish,
moving
good.
along.
What speed is Herc going at right now?
0.2.
0.2? Thank you.
0.2 knots. Oh, I'm on the sand.
Yeah, this would be a very sandy area.
They said we'd have to stop for a whale fall, and of course we would, but
Of
I think this is a
course we
little
would.
shallow for a whale fall.
I mean, if it just fell down
Like,
here.
it might roll down.
Yeah.
It's pretty steep.
Wouldn't have much to keep it staying. That would be cool.
Yes, that would rise to the occasion of, well, maybe we should stop and look at this.
Be another chemosynthetic ecosystem.
Jeff, they'd like to know if you've ever found a whale fall.
I personally have not. I've seen footage and read about whale falls. They sound very interesting. It's told of a story at Axial Seamount where there was a whale fall, and then the amount of spider crabs that came to eat the whale fall. So at Axial, you'll see a few spiders, every few hundred meters, or even maybe a kilometer. The density of spiders is not that much, but you do run into them.
Oh,
when a whale fall happens, they come from far and wide to
Spider crabs or sea spiders?
Spider crabs.
I can handle those.
Yeah, although they are large and I think they're a little scary.
There's some more coral. There you go.
Someone says Herc just kept swimming with all the amberjacks the other day.
Yeah.
Just keep swimming.
Just keep swimming. The
Just keep
amberjacks
swimming.
swam with us.
Yeah. They were like,
They wouldn't leave us alone.
It was kind of cool.
It was cool, yeah.
I'm wondering if that's corallium or just corallium down there. It's very pink.
Why did the whale fall? It was a fluke.
Oh no.
Oh no.
I don't know. They said it, not me.
I hear you.
We're
I
not
get
going
it.
to do this today, okay? We had our silly time yesterday. We got to tone it back.
Yes.
Tone it back.
Yes. But
Actually, Jeff and Kelsey gave really good lessons yesterday.
They did.
We filled the blue water up. The blue water time.
Yeah, I remember saying nothing of consequence, but I'm so glad you do.
You described all the transects
We
and
know
everything.
what
You
you're
taught
doing.
a lot yesterday.
Yeah.
Maybe it was just sleep deprivation at that point.
It's all a blur.
It was a busy day yesterday.
You were in the control van for a long time.
It's honestly just like my office at NMC, which is no windows, hold up at a computer, you know?
I think I could probably deal with an office like this if it had giant monitors showing the seafloor.
Yeah.
I'd get no work done.
Oh, no.
That'd be too distracted.
This is work.
This is
Can
work.
you pinch all of us? This is work? My gosh.
This is pretty cool
We're
work.
very lucky.
I don't know. I don't think I can go back to my regular job after this.
No.
Let's hope my boss isn't listening.
It is tough to go back. Like, I could be out on an ROV dive right now.
There's a little fish floating around.
Going back to Whale Falls.
Yeah, tell me more.
Well, I'm just trying to think about if we would see one all the way out here. Because, of course, in order for there to be a whale fall, we have to be looking at, you know, areas where whales regularly use the areas as highways, right, of the sea. And there was actually a really cool paper that was published in 2020. And they had found a new breeding ground for humpback whales in the Marianas.
Oh.
And so I'm pulling up the paper now to look on the map where they had found the observations. I don't think they went this far west, but they did find them closer up to the archipelago. And in January and February, we actually do see humpback whales. Like I can see them off my porch as they're coming by, but it's
they go by Saipan, but they don't tend to come by Guam.
Yeah.
We get the pilot whales and some sperm whales, and every so often we'll get a little whale shark. So all of those are pretty rare. The pilots we do see every year, usually the sperm, but the whale sharks are probably every 7 to 10 years.
Wow.
I love whale sharks. They're so pretty.
Yeah, they're more favorite.
And apparently,
Sierra.
What?
They would like more magic tricks from you.
More magic tricks.
No.
No.
I'm going to have to make Marin work overtime over there if I want more of those.
Gotta come up with something
I
new.
had to remember what that was about. I'm like, what? Oh, yeah.
Yeah, there was a lot of...
There's another little brittle star down there.
I don't know how many dives there are left in this season. We have another 6 more days.
On this expedition.
Yes, on this expedition.
Yes, 6 days, I think possibly 5 dives.
That'd be nice.
6 days, 5 dives.
And then
depending on the schedule, they might have to go down to 4 dives. But yeah, and then what I think we're at, is this dive 2000 right now?
2100.
2100.
2100.
Dive 2100. So we've done pretty well on our expedition, and this is the first expedition of the season. So
yes.
How many dives are left in the season or the expedition? We got maybe 5 more dives.
Well, they did say how many more are left in the season. I really have no clue.
Hopefully.
I can only do this expedition, which ends on the 29th.
Yes, we'll be concluding
Back
our
and forth.
Mariana arc, back arc, remnant arc, and handing over to the next expedition that's going to go look at 4 arc and mud volcanoes.
That does sound pretty cool. I think I want to sneak my way into that expedition.
Just don't get off the boat.
Just don't. Well, we only had 51 people out of 52 on this. I could have just, you know, if it's the same on the next one, just kind of stay. So yes, the Nautilus does travel around to different areas of the globe. Right now we are in the Pacific. They tend to do their expeditions, kind of clump them around areas of the world. So you never know where they're going to be. They are planning, usually are 2 to 3 years in advance, but all of that information gets released to everyone at the same time. So probably next March or April, you'll be able to find where they're going to the next year.
So it seems like we keep coming across a boulder every now and again, and that's where we're seeing the most kind of coral in life so far.
I'm kind of excited after the transect to zoom in a little bit to see if there's any little things in there. Doesn't seem like it, but...
Well, what we're not seeing is all the tiny little things that are in the sediment right now. All the myofauna. It's a pretty large group of diversity. We can't see it, but we got
not
some
even
samples.
If we zoomed in.
No.
We'd have to take a scoop or a suction sample and then look in a microscope.
Is that the plan on this dive?
I don't think we had planned to take any of those samples.
I didn't see the scoop packed on the ROV.
No scoop. Although we can use the suction sample if we wanted to get sediment.
True.
So what kind of infauna could you find around here?
In the sediment? You could probably find a lot of polychaetes and nematodes. They kind of make up the largest portion, so our worm friends. Probably larvae, so stages of like crustacean larvae or different kinds that stay in the sediment. Trying to think what other groups we could get. Sometimes you'll have water bears in the sediment. Those are pretty cute. Water bears are the organisms that can actually survive in space.
Oh.
Is that like the tardigrade?
Tardigrade, yeah.
Did
They
they
do look a
test
little bit like
that,
water bear,
or-
don't they?
Yeah, that's just another name for them.
Another name?
When you interview to be an SCF, one of the things that they asked in my particular interview was, if you could be one creature, what would you be? And I said a tardigrade because it can survive just about anywhere. So
How do they know it can survive in space?
they've brought it up to space.
Yeah, they've taken it.
Okay, that's what I was...
I think they've set them on fire and brought them into volcanoes and down to the bottom of the trench.
These poor tardigrades.
Yeah.
I didn't know that.
Yeah, they have this really cool like stasis mode that they can just like make their entire body like appear like they're dead and like freeze all function and then come back to life when they need to.
Whoa.
They're funky little dudes and they're cute.
They are really kind of cute.
Microscope.
They're kind of like a Michelin man. They got like big rolls and fluffy.
Combined with a mole.
Animal.
Yeah,
Animal.
like a little mole.
So someone asked how long this dive is planned to be. We're at 8 hours. And yes, you're correct that the next watch will be the ones on the top.
As you can tell, we didn't waste any time moving because I wanted us to get going on these waypoints so we could get up and see some stuff.
Yes.
To be fair, yesterday's dive, the deeper part was the cooler
The
part.
first part of the dive was the most...
get to the very top is too hard.
Yeah.
So count your blessings. Okay.
You never
never know.
You never know what part of the dive is going to be the coolest.
We're still getting to dive. I mean, this is the life.
Yeah.
And you never know, they may extend it. We've been, having that happen.
Honestly, it's such a privilege just to be able to be out here in the ocean.
Couldn't agree more, Annabelle.
Absolutely correct.
Another boulder.
Nope. Will this boulder
have coral? Oh.
How far into the transect are we? Oh, I can look on the iPad.
We're a little over halfway.
Yeah, nothing there. If we do see those really pink and white precious corals between transects. It would be nice to get a small snippet.
Pink and white?
Yeah, we don't have those yet. And I'm thinking it's either a corallium or a pleurocorallium.
Okay.
Which are they're called precious corals because.
Do we want to mark this area?
If I see another one.
Okay.
Yeah, I have a feeling we might see some more though further up.
Okay.
So they're really called precious corals?
They are, and it's actually because they used to be a fish species. So there was a fishery for these coral around Hawaii because people turn them into jewelry. But they are very vulnerable, and they are one of the slow-growing corals, so they take a lot of time to bounce back if they're overfished. So now we don't have those have been closed because we've been kind of seeing that by taking up too many, there's not enough left to recover. So now it's a very kind of more frowned upon practice. But there is some cultural value to precious coral as well.
Oh, what is that?
Urchin.
Oh, okay. This has a very different geology than what we've been seeing. It's got a couple of different types of things all in the same area.
Yeah, we saw a lot of like this carbonate rock. This rubble, we didn't see as much, but I think it's just because we're getting steeper that there's more shedding from probably the reef up shallower coming down.
Look at that sonar.
What kind of slope are we traversing right now? It's pretty steep. Pretty steep.
Look at the Atalanta cam.
Yeah.
Gonna have to start moving up.
And then if you look up at the sonar.
Yeah.
The wall.
Yeah, sonar's pretty bright. At least it doesn't have that like right angle from the other day. Oh, there's another one.
Yeah, I'm looking for the bright pink and white ones though.
I just like that it's called precious coral.
Precious, they are very precious, and they're all pink or white.
They're
it's actually they're called a fishery.
Yeah.
Even though they're not consumed.
Yeah.
They're eaten. They're not eaten.
They're not eaten,
but
for
they're
goods.
Still fished.
They're like mined almost.
Yeah, but because they're an organism.
Yeah.
Yeah, it's a fishery. There's some really interesting conversations that have been happening on Saipan because a clam shell that is used for cultural practices for jewelry making has been proposed to be put on the endangered species list. Federally, so on the ESA. And one of the things that folks are really concerned about is if this gets put on the ESA, how will one know, if it is a shell that they have already had or had been gifted to them, or if they are an artist, and that is their livelihood. So it's really interesting when these species that have significant cultural import to the communities then get put on different federal lists and the conversations that arise from that.
Right. Been a big topic of conversation in Guam too, because it really is cultural to have one of the necklaces and how they're given and seen in the islands. I think this is very similar to many, many, many years ago before I moved to Guam, when they put sea turtle on the endangered list.
Whoa.
I'm glad it went on the endangered list. Ooh.
Look at that sea star.
Yeah. But it made it so you couldn't have any of the shells or the jewelry or any of that. How many arms does that have?
Let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9. I think 10.
Huh.
10 arms? I don't know what kind of sea star that is, though.
I can imagine it gets tricky when the intersection of conservation and culture.
We have some really interesting conversations in the classroom. As Shannon said, the Sanahi, or half moon, is really important. It is gifted and has a lot of symbolism. And so that is where we have need to have really strong representation and communication when federal regulations are being considered.
Are they fished for anything else other than the
clams?
The clams?
They're fished for their shell and their
to
meat.
eat them?
Yeah, yeah.
And honestly, there aren't that many left on island. I mean, we do have some, but it has diminished. But when one is found, it is used culturally. It doesn't go to waste.
There's another little sea star, maybe? Or is that a squat lobster?
Looks like a lobster.
That looks like a
very
really long arm squat lobster.
Yeah, chirostylid maybe.
Speaking of cultural, our expedition has a Chamorro name, right?
It does.
Matingan.
Matingan.
Matingan means deep water, ocean water, not just around the island, which is the reef water. It is actually going out farther and deeper.
It's a good name for this expedition.
Very good
We have
name.
done that.
Does anyone know the next expedition that's in the CNMI, what its name is?
It's a Rapala wash name. I'll see if I can find it.
Yeah,
I have it somewhere
thank
in
you.
my notes.
We have one of the scientists in the chat saying that around the Marianas, ooh, fish.
They're back.
It's close to a very famous precious coral fishery area called Ogasawara, and that there has actually been some illegal fishing that happened years ago.
Yeah, 2014.
So it could explain why we are seeing some of these precious coral species pretty close by.
Based on that name, I'm guessing it was a Japanese named site.
Probably to the north, and yeah, some.
In the Ogasawara region. Oh.
Right.
So yes, this expedition does have a cultural liaison. I think that's what we would call Sean.
Cultural, and he's representing the monument.
Yeah,
he's the director of the.
He's the superintendent of the Mariana Trench Marine. So he's actually the resource permit monitor. Now, having a cultural liaison on board is actually something that OET has been discussing for next summer's cruises.
Oh.
But I will say one thing that OET does do very well is try to work with the community and make sure that folks from the islands where the exploration is occurring to have folks from these communities on board.
Sean has been invaluable on this mission. He is always comes up to the van when there's anything, anything and everything that he has knowledge on, which is pretty diverse.
He used to work for the local Division of Fish and Wildlife, and then went to go get his PhD at University of Hawaii, and then came back.
The transect. But keep up the conversation though. This is really interesting.
And when his prior at DFW, his specialty was food fish. So that's why he's so knowledgeable regarding all of the fish. But it's also fantastic because he has not only the scientific name, but also the Chamorro name.
I have the transect segments. You guys want them now?
The
transect done?
Transect done, yeah.
Transect done.
Transect completed. We get the sequence numbers and.
It is 4, 5, and 6.
Awesome. Thank you so much.
Yeah.
Okay, well, we're now free to stop, look, zoom, collect, and keep an eye out, although we're getting steeper. Keep moving on to waypoint 4, but we can we can rotate and look around off to the sides if so our field of view gives us a chance if there's some interesting things off.
Thank you, Mike and Marin, for keeping that steady and zoomed in.
I think
that's the gauges.
Roger.
Isabella, how shallow is the top of this seamount?
Oh, good question.
Oh, sorry.
No apologies necessary. Shucks.
Well, we're getting a depth at least
I think that
65 meters, but I think it's shallower.
I don't know what our shallowest sounding was yesterday. I don't know if we went over like the shallowest, but I think there might be like a 21 meter.
There's some on the nautical chart.
Yes.
Out here, it gets into the 20s. Maybe shallower.
But
so you're telling me that when we're about to recover that I could just jump off and find some scuba gear and see what's here?
Probably not where we're recovering. Yeah. You'd have to swim a ways away to get to the shallowest bits. But possible, you're in the big scheme of things pretty close.
If I'm the first one to scuba dive on it, do I get to name it?
I don't think so.
I don't know. Sure.
Jeff's like, whatever you want, sure.
Just be aware there might be sharks, and the equipment comes first, so.
Whoa.
The equipment comes first.
When did that get put out there?
I'm okay with sharks, well.
Equipment
maybe not first. That like oceanic white tip we saw the other day.
Jumping in the middle of an ROV recovery.
I think we still got to go save her.
Equipment does come first.
What?
No,
it doesn't.
Yeah, you guys, come on.
Don't spread these our priorities.
People come first.
It says life and property. Life is first.
And for some reason, Shannon is in the water. We would get Shannon first.
Well, it would be a, hey, Shannon, why'd you do that? Oh, you're going to go swimming? No, you're going to get back on the boat.
I would never be allowed in another expedition again.
No, you would be banned.
You just had a calling. You had to see the water.
The water was
train looks good.
You do get people that have the calling of the void, which is like a psychological thing where, like, staring out into the darkness, there's like, oh, should I jump? Should I go swimming?
The darkness stares back.
The darkness calls back.
Well, we are about 390 meters, and the water is 12 degrees Celsius.
Chilly, but not as cold as it has been.
Oh, yeah. That's better.
I'm not seeing a whole lot so far on this side.
Yes.
Could be a weird transition between areas. Pretty steep though.
I'm amazed we're already at between waypoint 3 and 4.
Right?
We're going a lot faster than I anticipated.
How's the current? Are we experiencing any currents at this depth?
There was some out of the when we started. Out of
red up there?
But there's not much.
Oh,
can we see that?
Take the sky.
Little starfish. Oh, there's an anemone too.
There was a little bit at the start, but pretty.
Not much. I'll just have a look here now after we get up, but not much to talk about.
Okay.
Great.
That makes driving and getting around a little easier.
Yep.
This guy doesn't have much of a home. It's just that little rock hanging out
What is
above
it?
him. It's a starfish.
Here, go ahead, zoom in.
He's just holding on. How did I get here?
Whoa, look how bumpy he is.
That's kind of cool.
Thank you.
And pokey, too.
Yeah, I was going to say, he's got those little hairs.
I have not seen a starfish like that. That's awesome.
Getting closer.
Ooh, is this something we...
I don't know.
Do we know what kind it is?
Not off the top of my head.
Looking
It looks a
up.
little
I think
bit
I
like
found one.
olives.
Looks a little bit like olives?
Yeah.
Yeah. On a toothpick
Yeah.
over a martini glass.
All right, we know where your mind's at. Shaken,
Okay.
not stirred.
It looks like a Coronaster briaris. Boasts
Coronaster briaris.
10 to 11 arms. I'm not sure if this is the correct one though, because it says this is in the Mediterranean, but it has the same bumps.
Oh, I see what you're looking at. Yeah.
So it could be related, but it also has the same bumps and kind of the spines coming out.
Yeah, this
Hello from the
yes,
from the
hello.
lounge.
Eva, yes.
It is a coronaster.
I got it right?
Is it
Yeah.
a species?
I don't know about the species. But yeah, then they're always like underneath a crevice like that, so they look like just a weird amorphous blob.
Amazing. So have you seen the coronasters in the Pacific before?
I've never seen them before, but I know they are known from around here.
Okay.
Got it.
I'm in the I'm looking at the benthic Deepwater Guide, and they have been seen in the Mariana region around 350.
Which is we're at 384 right
Which
now.
is, yeah,
It's about right.
it's about
Amazing.
right.
And this
Thank
one-
you,
yeah.
Eva.
Thank you, Eva.
And we had an urchin
You're
off to
welcome.
the left,
We're
I
right on
think.
the 20 right now.
Or the anemone on the
Is
left,
it?
yeah.
Oh, an anemone, sorry.
It's okay, though.
We're,
We gotta catch up.
that was a good spot.
Good
He
spot.
was
Yeah.
neat looking. Oh yeah, that's
That's crazy.
that's it right there.
I'll put it down in the notes, maybe.
Kind of looks like grapes.
Or
Like
olives.
or olives.
I think one thing that's important to note is it's very hard to get to species level, simply because you need to do genetic testing. Also, just from images, you're not able to look at all of the different morphology context. So it is safer to provide a genus than try to always provide a genus and species level, because that level of refinement isn't always going to be accurate in the field. So
Very
the
true.
genus of this is Coronaster, as was confirmed by Eva and
Oh, looks like
Zero.
there's another one on the right, or on the left there too.
Oh yeah.
Good spot. Is that the same kind?
They're kind of just like plopped. There.
Yes.
Go ahead and zoom in.
Got it?
No, it's
The
same
Mariana
kind.
Trench, the subduction was initiated at the Mariana
Yeah.
Trench about 50 million years ago. So there's 50. So 48 to 50 million years ago, subduction initiated, and there was a long history of subduction occurring without back arc spreading, or, well, and then they had intermittent periods of back arc spreading that created these remnant
Okay,
arcs.
full wide.
But
Got
the
it.
initiation of the Izzy-Bonin Mariana subduction zone system was about 50 million years ago.
Ooh.
Yeah.
Pull up a bit more, Annabelle.
Roger, coming up.
Jeff giving a shout out on Ship to Shores.
Yeah, just turn around. There they are.
Oh, so there's only 2 species within this genus. It's a very small group.
So what sort of advantage would a sea star have to have those weird little olives
That's a good
on its
question.
skin?
Maybe you look less appetizing? I don't know. Yeah, it's interesting
There's
that
a
they're
ton of
all
them.
found just like,
What is
there's
that?
another one.
Right there, yeah.
Yep.
So obviously we have them, I guess that is thriving here if we're seeing.
Livable conditions.
I'm amazed we don't see anything else around it. At the beginning we were having several species of things together in little pockets.
Yeah. Well, don't know. Get into different water masses as we get up closer to the photic zone, to the mesophotic zone.
Sierra, is it common with deep sea species to only have one per genus?
It's very variable. It just depends on the group. So some can be like Kelsey was saying that you really need a good sample. There's might be another one down there.
Another one?
If there are a bunch of in that group, then it's only really able to
Can we
tell
look
them
at that
apart
over here?
morphologically
Sure.
or genetically. So
It
you do
looks
need a
like-
sample
Is that
to identify it.
another one?
I
or
didn't
is
want
it-
to say it, guys, but it looks
No,
like
I thought it was an
moving.
octopus.
Is it an
Nice.
octopus?
That's what I was worried
I
we were
was
going to
holding
pass
the
by
word
the
back.
octopus.
I did see it and be like, hey, didn't know it was an octopus, but
Good spot.
ooh, of course you know you're zooming.
Guess who just said earlier that
do
we
you want me to
needed
get
an
the lasers?
octopus?
We needed an octopus. We
Sure,
would
yeah.
have stopped on a transect for this.
We would, yeah. That's pretty worth it.
Oh, he blends in really nice. He's
Oh,
going in the
what?
hidey hole.
Oh.
Oh, well, he's shy.
Too soon. Oh, that was so cute.
Well, we
Was
saw
it
him.
too quick
She
to
kind of
get
saw
a
it go in there too. That's kinda
identification?
Yeah, I couldn't get a close picture of it.
cool. What awesome habitat though.
Yeah.
Yeah.
Just this little carbonate hole.
How many octopus are out there that we just don't see?
True.
that are all.
That is exactly what I was thinking.
Someone's going to be annotating the transect
Okay.
footage later and be like, there's an
For what?
octopus.
Okay, well, we
Got it.
can continue. I think he's going to stay in there.
He's going to hide
We're
from
sorry to bother you.
Oh.
We just wanted to take your picture.
I guess we can be on the lookout for more.
Yeah, good spot. He was, as soon as he saw us, he was moving.
He was not happy with
What's
her.
up in here?
Yeah, there's
Oh.
something in that one too.
Perchance another, is it an eel?
Maybe an eel.
Go ahead and zoom
We
in.
have been seeing eels
Got
in
it.
the area.
Or more
Is it a
like a fish?
Let's see. Oh, I can't see his little head.
Come on out. Come out.
Said no. Oh, is
You
it
can
a
see
little
us all
eye?
leaned into our screens.
Oh.
He's kind of
Getting
pulsating
closer.
back and
Coming
forth a little bit. Ooh,
It's a fishy.
Little fishy
Yep.
fishy.
he's like hovering.
Okay, full wide.
Got it.
Cool. Very cool. So is 4 to 5 a transect, or is it 5 to 6?
4 to 5 is a transect.
Okay, well we're coming up on another transect, and that might be a good one.
Could get some cool animals. That was awesome.
It like tucked away in that little hole. It was so cute.
Yeah.
Ooh, I guess the feed was glitching when the octopus showed up.
Oh,
Well, we only saw
no.
it briefly.
Yeah.
We barely even saw it. Yeah, sometimes the animals will cooperate, and sometimes, you know, they see a giant Jeep-sized vehicle with bright lights coming at them, and they do what I would do and go and hide in the hole.
Can't blame him.
Can't blame them.
Marin, I guess there's, I don't know, but a lot of people are saying there's audio-visual issues on channel satellite 1. I can't tell if it was short or long, but someone's saying the cameras aren't working.
On my end, everything is working.
Yeah,
so,
I see it up on mine.
Yeah. Might just be a connection issue on their end or something.
There's
Yeah.
another, that might be another one of the sea stars down there.
Thank you for checking on that.
I'll keep an extra eye on it too, so thank you.
Thanks.
Go ahead and zoom in.
Yeah, he's just in his
Got
little
it.
lump.
Yeah, they just like wrap around these.
They are really cool how they just...
Oh, and there's a little coral down here too. See his little polyp?
Where?
Right down here.
Oh yeah, it's yellow.
Little yellow coral.
Can we zoom in and see it just a little bit? Or is that too close?
I think that's fine.
Is that all the zoom you have?
Oh, I wasn't sure which one. Yeah.
There you go.
Ooh.
Oh yeah, it's got like 2 little polyps on it.
Thank
Oh.
you.
That rock looks like dentures. Cute.
Well, it looks
Okay,
like some
full
kind of
wide.
scleric
Got it.
canyon, like the
Our
start
transect
of
bearing
one.
will be 010 pilots.
Roger.
010.
We're still 20 meters off the beginning of the transect.
Roger.
So when you have your mom listening to your feed and she tells me not to jump off the ship with scuba gear.
Oh, that's a good mom right there.
It's a good mom. Sierra, we haven't seen your dad on in a little bit.
Oh, it's probably, let's see what time it is in Arkansas.
Going to be like 10?
It's like 10:30, yeah. It's probably bedtime. Shout out Eddie Landreth, my father, for tuning in to some of these. He always sends the best questions.
Yes, he does. Sometimes we don't know till the next day.
Yeah. Some more kind of yellow coral looking.
It's a little bit of a discoloration there.
Might be like yesterday when we start seeing those like little specks of like yellow and purple and
There's, what did they call it? A 1980s mall?
there's maybe another sea star right there. They're just all
There's
in
something
this area.
purple or blue before it.
Yeah, might be some. We might start seeing some crestoise coralline algae when we get shallower. Or maybe down here.
All right. Transect 2 is started.
Excellent. What's
Let's
the
get it.
time?
The timer, the
Oh, 3,
UTC?
4. I got it.
Yeah.
And then the segment?
we're starting with 7.
We are back in transect mode.
And then
Yes.
depth?
So we are in transect mode. There will be no zooming in. For the next 150 meters, we will be staying
Like 330.
steady
330, thank you.
distance to the bottom as well as video.
Unless we see another octopus.
What
Thank
is
you,
that
team.
yellow thing? Is that coral?
Might be. See some polyps.
Thanks nav, thanks pilot, thanks video, thanks data logger. Appreciate you all.
We're gonna get these transects.
Thank you,
Going.
Kelsey.
Do we get this? Is this the last one today? There's only 2?
Yes.
Yes.
Okay.
I might see if 4 to 8 can do one, but for 12 to 4, this is the last transect.
Gotcha. Starting to see some more color.
Whatever that orange thing is down there, too.
Yeah, might be another one of those.
Ooh.
Ooh, look at that. Oh my gosh. It's a crinoid.
That's pretty.
It's one of those ones with like the
We're in transect.
transect mode, yeah.
Can't stop. Yeah, we did get it.
I took
In
a
the
picture
transect.
of it.
Yeah.
And we know what that is, so that's
Yes.
good. We're in the transect.
I've never seen one of those before, though. That's so cool.
Annabelle, is it alright if I turn down the iris on?
Yeah, sure.
A little bit. Cool.
Yeah, we are glitching on all of our channels.
Are multiple people saying that, or is it?
People are saying that in the
People are saying
chat.
it, and I'm on the site now.
Oh.
That might not be helping.
It seems like something internet related, because even my internet on
Video, do you want me to message Ed?
Yeah, if you
yeah,
could.
please.
Thank you.
Trying to pull up the
Don't know if that's an
species,
Ed
or
or
at least the genus.
Michael thing.
Yeah, so that was a metacrinus or a serocrinus. I will write that down. Good spot.
So science, what's so important about a transect? What can
Hey,
this
Annabelle,
kind of data collection?
could you move your a little closer? I've been having trouble hearing you.
Sorry about that.
Oh, there you go.
Can everybody hear me?
Yeah, you're sounding good
Yes,
now.
much better. Thank you.
Awesome. All right. So what's so important about a transect? So like how can collecting data this way help you in your studies?
Well, one of the ways that this is helpful is if you are collecting at the same depth and for the same duration of time and along the same length of transect across multiple different sites, you can do comparative analyses. So because we have relatively similar depths from yesterday and today, we have the same length, we have the same speed of capture, and so we're not duplicating how often we might see another organism in the transect. We're able to compare these two seamounts. Transects are a way of evaluating a whole bunch of different variables, from biodiversity of organisms to, you can look at benthic composition, which is what we'll be looking at. You can run transects to count fish and do fish assemblages. That's one way. Another way is called a point count. And you can also do this terrestrially as well. So you can walk a transect and, you know, count how many sorts of your target species you encounter from that transect. So it's just a repeatable and methodical mechanism for collecting data. And one of the things about science is its repeatability. So by knowing exactly where we started and ended these transects, in theory, anybody with an ROV could come back out and do these transects over again in time, and then you would have a temporal data set as well. So you'd be able to compare not only in geographic locations, but you'd be able to compare a site and how it has changed or not over time.
Awesome.
That's so cool. Thank you so much for sharing.
Shannon, Ed said something about submitting a video down report.
Okay. Yep, I just did it. Thank you for making sure I knew that.
You are most welcome. Transects are the bread and butter of marine biology.
Is a video down report result in something happening? Or is it just a report?
I don't know.
Is that like a TPS report?
Might be a report that sends to the data engineer or something. But that's just my guess. There's something on
I've seen a lot of little...
Was that on the right, Sierra? Was that like a sponge or?
This looks like, oh, the big white thing? Yeah, that might have been a sponge. There's a little sea fan over here. These are some coral, it looks like, on the side.
We have a lot of scientists on the back row. Do you all have any other, anything else to add about the usefulness of transects?
I like them because if you get replicates, you can do things like averages. So a lot of times, you know, seamounts and these kind of habitats, you might have an area right next to each other that you're seeing a lot, and then the next area not as much. So if you have, you know, replicates like, for example, 3 different of the same distance, you can kind of get an average over, you know, a wider distance, which is kind of cool. I use them a lot to look at community structure and composition in different areas. So it's really useful if you want to compare locations or depths. Lots of different variables and questions you could answer with transects.
Yeah, transects are like when you're setting up an experiment, you're trying to control as many variables as possible, and a transect
Are these just rocks or
allows
are
you
these something
to
with eyes? Sorry,
say
Jeff.
I did this at this depth in this path and kept this view, which is one way to do it again. If you want to do the temporal, analysis. There is something to be said about the random walk, which is kind of the antithesis of a transect, would be the random search. And if you do a large enough random search, you can get a sense of the area, but it's difficult to reproduce a random walk inherently because it's random. So there's trade-offs because the transect is great for that reproducibility, but you are looking in one narrow view up on the transect. So you have to make some assumptions whether or not your transect is representative of the area if you start wanting to make interpretations beyond what you saw in your transect.
Exactly, and that goes for if we were to really want to get a representative sample of this particular seamount, we wouldn't just do 2 transects. Right. We would do multiple transects across and around the seamount, across all of the different depth contours. So, but that would take forever.
There's trade-offs to any sampling scheme with availability and time and what your objectives are.
Right. Exactly, and so that goes to the importance of shaping a research question and trying to identify what kind of question you're trying to answer. And then your research design and methodology is all geared to then answering your research question. So the research design component is actually one of the most important parts of the scientific method, and I think it is oftentimes overlooked, and then you get to the end and you're not actually able to do the analyses that you were hoping to do because not enough time was put into the original research design. And that's just a common problem. Sometimes, you don't learn these things until you've gone through them.
Just to let our audience know, we are aware that there is some issues with our live stream. It has been reported and is being worked on, so hopefully it'll improve a little bit. So what is all this yellow?
We kind of saw this yesterday. It's
Can we open the
some
iris
staining maybe, or
Thank you.
bacteria?
Encrusted growth.
Or encrusted
That's
growth.
perfect.
I've been seeing
Thanks,
these
video.
little white bumps with holes, and I'm wondering if these are some coral or if they're something else. They don't look like...
When we got to the shallower parts of the dive yesterday, we had the like the red and green color on the carbonate.
They called it neon glow, I think.
The neon glow, yeah. Neon I wonder if this is the first bits of it, and then it slowly just gets more and more, and then you get the red comes in.
Right.
I'm not sure.
That could be. I'm curious what these.
We didn't see it down at lower depths as much.
Right.
Rebecca Carey. Hi, Rebecca. In the chat is saying interesting substrate. Is this all carbonate?
That's almost certainly, yes, all carbonate.
Yeah.
The rock that was picked up yesterday at the same kind of environment was a carbonate rock when they cut it on the rock saw.
I'm so glad they cut it. It's pretty cool inside. I know they don't cut all of them while on board. What are those? Are those coral or what are those dark spots?
I don't know. These look like coral to me, but I'm curious what these.
We can't zoom in though.
Yeah,
no, I know we can't zoom in.
Because they all seem to have these like 2 holes in the top of it.
Like
yeah,
eyes.
It's interesting.
I don't know.
And we have some of those blue things too.
Almost kind of looks like a clam.
Ah.
Huh. Oh, something just moved.
Mm.
Right about where the lasers are right now.
Oh.
Oh, maybe in that hole?
Yeah, to the hole on the right.
Okay.
Oh, yeah, maybe a little poking out.
Yeah, these are little sea fans.
It looks like Atalanta is right at the halfway point, and we are just a little past the halfway point based on the high pack.
Roger.
Doing great.
Mm. Well, you'll have lots of things in this transect to be able to extract data from. I mean, lots of coral and...
They're nicely spaced out, too.
Yeah, they are.
They're a nice distribution.
It's
Not a lot of stuff on top of each other.
Another little urchin, I think.
It's also a fantastic training set for students as well, just because you're not overwhelmed with the abundance sometimes when you're doing benthic assemblage on coral reefs in the shallows. There's just so many different types of corals, especially in the Marianas. We have over 376 different species of coral in the photic zone. And it can be very overwhelming when you're first trying to tackle learning how to do percent cover.
I imagine one of the things to learn is that it's okay that it takes a long time.
Yeah. It depends on who your advisor is.
No, that's true.
Yeah.
Yeah.
I will teach them that.
To do it,
yeah.
it's better to be slow and get all the species than to go fast and miss stuff. At least that's what I think.
Yeah.
I'm looking at our map, and we're a lot more northwest than I thought.
Yeah, we are. We're quite northwest.
Yeah.
Right.
Yeah.
I thought we were getting a little bit closer to Guam.
Not yet.
Not yet.
We're working our way back, starting tomorrow, heading towards that back arc site.
Mm-hmm.
It's back arc and then moving south.
So we've come up from about 550 meters at the beginning till about 270 meters now. And the temperature has increased. We're at 19.5 degrees Celsius.
Oh, we got some fish.
Ooh.
That's good for the transect.
Those are the gindai.
Gindai. How do you say it?
Gindai?
Oh.
G-I-N-D-A-I.
Gindai.
Everyone's got a little
They all
hidey
have
hole here.
great hidey holes.
Yeah.
Yeah.
You know, we're not seeing those trails like we saw yesterday.
I think you can see this whole thing is about sliding off.
Yeah.
No, not as many trails. Or
whatever those trails were.
Well, I think they were fractures, like breaking up of the carbonate and then filled with sediment. And they look like little trails. Kind of like this, but smaller.
Right.
You have this too, like these are just little fractures coming down on the rock.
I'm fascinated by those little holy critters.
Yeah, part of me is wondering if they're tunicates.
I think that they are tunicates.
What are tunicates?
They are technically a type of their hemichordates, right? So they have partial notochord. So they're technically not, well, they should be, they're not vertebrates, sorry. So, but they are chordates, but they're very squishy. They're almost like salps, so they can sometimes be soft, but sometimes I guess they can be hard too. Some attached to the seafloor, some are free floating in the water. There's something cool right over there. I know we can't stop, but.
Got a fish.
Another fish. But yeah, these ones look to be more colonial to me. You've got like this whole bunch right here.
Oh.
Oh, wow. Yeah, that's cool.
And they're not moving at all.
No, they're sessile. They're stuck to the
We're
substrate.
almost at a point where we can go in and look closer.
We're about 25 meters away from the waypoint, I'm estimating.
Oh, that's pretty good.
Right?
This is a nice transect.
Yeah, it is.
You're going to have a lot of coral.
Yeah.
And tunicates.
I wonder how many coral that is.
Coral
So
plus
many.
plus.
So Gindai, by the way, is just the English common name, but the scientific name for them is Pristopomoides zonatus.
It's a pretty name.
I want to give just an update if you're watching or just tuned in. There is some video issues happening right now. The video issues reports have been issued to the people that can hopefully fix it, and they're working on it. But apologies. And just another update, yeah, we're here at this unnamed seamount in the western Mariana Ridge, going up the slope on the south side, and we're moving pretty quickly. Some of our learnings from yesterday says get shallow if you can, where hopefully there's more abundance of species and more biodiversity. And if we get to the top and there's a lot of stuff going on, we'll probably hang out most of the dive near the top or on top of the seamount.
Wow, so many more of those corals. It's pretty cool to see.
This is an 8-hour dive.
Yes.
So we don't have a bunch of time at the
and we're 2 hours into it.
2 hours in,
dive.
so there's 6 more hours of this dive left.
I'm wondering if those very specific circles are evidence of like predation or presence of an urchin at some point. And it was
Yeah,
just
they're very circular.
Could be. Good notice. Good spot.
Now we just need to see an urchin and like
Confirm.
I saw one earlier. It was kind of like it looks as if it was at one point a disc and then puffed up.
Video, can we get a zoom on the Atalanta can?
Yeah.
Coral fields, aplenty. Might be worth to sample one of these guys.
Yeah.
Just so we can help our friends that'll be annotating later.
Yeah, I think that's a fantastic idea, Sierra.
Let's do it. Once we hit waypoint 5, we can stop and do a sampling stop.
Yeah.
All right.
Just because there's so many, I don't think sampling one is gonna.
Yeah.
There's a cool little black coral probably.
Oh, that whip, yeah.
Yeah, sea whip.
I always like when I'm scuba diving to look at sea whips to see what shrimp or little things are on it.
There's a guy floating.
The other thing that's interesting is they are all growing in the same orientation. So they are tip to tip, upslope, downslope, and then their fan component is actually like cross slope.
And we don't have much of a current here, right, Mike?
Yeah.
Not so much at the moment.
Oh, there's another. That's a Stichopathes type of black coral. We saw those yesterday too.
Was a Stichopathes requested on the sampling?
Black coral was requested. These would be technically black coral, yes. So we could honestly grab one of those as well if we're in a good area.
Okay.
These are a little bit shallower water, but still cool.
10 meters out from our end of transect too.
Roger.
Yeah, it's interesting, like you said, they're all just kind of that way. I feel like there's, typically coral would situate themselves in a way to get the most food possible. So maybe at one time there was like a current kind of coming this way or the other way.
It could be the dominant current direction.
Yeah.
A long contour.
Can they change their orientation, or are they just stuck there once they're tilted that direction?
The branching itself, the skeletal doesn't really move, but the polyps do have some ability to move their like tentacles. So the tentacles could move like left or right, but they're kind of situated how they are. They can't really turn that because they have a hard skeleton. It's not soft.
They're not like a tree that can grow towards the food source?
I mean, they could grow, yeah. It would take a long time, but
well.
it's a slow-growing coral. I don't know about these kind of intermediate ones here. These are a little bit more shallow water. The coral crop is plentiful, though.
Mm-hmm. It's going to be a lot of counting on this transect.
All right, that seems to be the end of transect 2.
Transect 2 over. We can park for some sampling.
Okay.
Yeah, maybe get an up close and then some samples.
Oh, what's that one right there? That's something different.
We get a zoom in on that.
Is that the end of the transect?
End of the transect.
Oh, and one of